Good morning, students. Today we will talk about the plasma membrane. In the previous turn, I have introduced you the cell, different type of cells, and cell organelles. So today we will talk about the outermost or the limiting boundary of the cell that we know as plasma membrane. This plasma membrane, when we think about the plasma membrane, the term cell membrane was given by Naseli and Kramer 1855. Now, what is the location or position of this plasma membrane? Recall the cell, either plant cell, animal cell, or bacterial cell. The protoplast is being guarded or limited by the outermost membranous structure. That is, this is the membranous structure outer to protoplast. This is thin, 70 to 100 angstrom in thickness, elastic, semi-permeable, that is it permits some materials to pass through it. It means it is selective permeable. Trilaminar means three-layered. Lipoproteinaceous means it is made up of lipid and protein, and living membrane. The second characteristic feature that we could see in the case of plasma membrane, that is when we are having the interest that what is its position, then it is between the cell wall and cytoplasm in case of bacteria plus plant cell, and as outermost boundary in case of animal cell. So, in order to know the structure and function of the plasma membrane, which is the outermost boundary of any cell or outer to the protoplast, what is the cell type that could be selected in order to study its structure and function in detail. So this is the mammalian cells or erythrocytes, RBCs, that is widely used for their isolation and further studies. If we are having the isolated plasma membrane, then via different chemical and physical techniques we could study its chemical composition, its structure, and its function in detail. So let us proceed one by one. After its isolation and chemical analysis and different physical methods, we are knowing that the chemical composition of the plasma membrane shows that this is made up of lipid and protein, mainly with a smaller fraction, that is 1 to 5% of oligosaccharides, and that oligosaccharides may be either attached with lipid, glycolipid, or protein as glycoprotein. Now this lipid and protein ratio is variable in case of different plasma membranes. Myelin is an exception which has dominant lipid than the protein, while other cells membrane have higher protein than lipid in lipid protein ratio. The main lipid of plasma membrane are phospholipids, cholesterol, and glycolipids. Based on the location in the biological membrane, the proteins may be 20 to 70%, and this may be of two types. Transmembrane proteins, when it is extended through the lipid bilayer, projecting out on either side of the membrane, not easily separated from membrane. So this is called intrinsic proteins, and this acts as channels, pores, and message transfer and for message transfers. The second category of protein is extrinsic proteins or peripheral proteins, present on outer and inner both surfaces of lipid bilayer. This is 20 to 30% with loose connections. Example, cytochrome C. So now, in order to understand that what is the location of this lipid and protein in the plasma membrane, various models has been suggested that could support its ultrastructure and different functions further. We will discuss different models that are being proposed and studied timely, who could be helpful in order to understand the ultrastructure of membrane and the function of membranes in detail. The first model we are having with us is Gotter and Grendel model, which was proposed in 1925, and this was studied in mammalian RBCs, and this model was having only two lipid layers. It means the membrane, according to this model, the membrane was supposed to be composed of two lipid layer only, and this was having the inner or central hydrophobic non-polar ends, while outer hydrophilic or polar ends. So, according to this model, if the plasma membrane is composed of only two lipid layers, then how could be possibility of ionic materials, passes of the ionic materials through this membrane? So this membrane was rejected. So the according to this model, cell membrane has bimolecular lipid sheet only when measured in RBCs, and it was with hydrophilic polar ends towards outside and hydrophobic and non-polar ends towards inner side. So what was the drawback of this model? This model has suggested that plasma membrane is having only lipid. So is there any possibility for the ionic material to pass through this lipid layer only? So this model was rejected. The second model being suggested at that time was or sandwich model or bilayer model or trilaminar layer, and it was proposed by Danieli and Dawson 1935. According to this model, the plasma membrane is composed of plasma membrane was 6 nanometer thick, and it was composed of the outer globular protein and inner globular protein, the outer globular protein and inner globular protein, which are towards the two peripheral ends of the plasma membrane, outer and inner. The thickness of the outer and inner proteinaceous layer was 1 nanometer, while the thickness of the central middle bimolecular layer of the lipid was 6 nanometer. So the collective or the complete thickness of the plasma membrane, according to sandwich model, bilayer or terminal model of Danieli and Dawson, was 6 nanometer in thickness. So according to this model, there was an outer and inner layer of protein, globular protein, and both were 1 nanometer in thickness, along with the central bimolecular layer of the protein, which was having in turn the hydrophilic polar outer end and the hydrophobic non-polar central end. So according to this model, lipid bilayer, the plasma membrane has a lipid bilayer and a protein layer on both the side of polar ends of the lipid bilayer molecule. The proteins on both the sides shows variations in different modified plasma membranes. Thank you.